Um, my name is Abena Dinizulu, and welcome to the NYU Langone Alzheimer's Disease Research Center's Learn at Home series. Today, Dr. Masurkar will be presenting on a talk on understanding the aging mind, discoveries from the Alzheimer's Disease Research Center study. Please note that we will be recording this session. You are muted upon entry. At the end of the presentation, we will have some time for questions. So you will have the opportunity to raise your hand and unmute yourself or pick questions in the Q&A box. So a little bit about our speaker. Dr. Masurkar is a physician scientist with a focus on the mechanisms, biomarkers, and therapeutics related to early Alzheimer's disease. Dr. Masurkar received both his MD as well as his PhD from Yale University and completed both his residency and fellowship at Columbia University. At NYU, Dr. Masurkar wears many hats. He's an associate professor of neurology and physiology and an attending neurologist at our Pearl Barlow Memory Center. He's also an investigator at the Neuroscience Institute and our clinical core leader at our Alzheimer's Disease Research Center. And with that introduction, I will turn it over to Dr. Masurkar, who will give us a presentation today.
Thank you very much, Abhinav, for that lovely introduction. I'm going to share my slides, and if just wanted to confirm with my team that it's being shared okay?
Yes, we can.
Okay, great. All right. Well, I'm really excited to be back on this platform to share the accomplishments of our center, of our Alzheimer's Disease Research Center here at NYU. I just wanted to go over the outline of my talk. I'll give a little bit of background about our center, and then I'm going to go over five key contributions, five key research areas that we have been working on in recent years that I think are very timely and would be of interest to you all, including subjective cognitive decline as a symptom of early Alzheimer's disease, the interaction between COVID and dementia, the application of proteomics as a technique to learn more about Alzheimer's disease, the relationship between blood vessel disease and dementia, and last, our efforts in the artificial intelligence space. So first, to give a little bit of background, our Alzheimer's Disease Research Center, or ADRC, is one of 37 such centers around the country, and we are part of a network, the National Alzheimer's Coordinating Center, or NAC network. And there's a picture of all the centers around the country to the right. And this network was established decades ago by the National Institutes of Health to facilitate collaborative research on Alzheimer's disease and other dementias. And over the decades, we have been collectively able to recruit participants, more than 55,000 participants across the country, and maintain a active, living, growing database of invaluable data. And these resources are freely available to all researchers. And we've also partnered via this data with other national consortia, really to answer very, very important questions in our field. And every center around the country is mandated to collect data in a uniform way so that we're all studying at the core some basic principles about Alzheimer's disease and its progression. But each center also has its own emphasis we can add our own flavor to the data that we collect. Now, our center is one of the oldest centers in the network. It's more than 30 years old. And in fact, our original cohort, the original study actually even predated the NAC network. It's over 40 years old. Our team is made up of several subgroups or cores, and that's pictorialized in the upper right, and that's the team to the bottom right. And each core is integral to various aspects of running the center, whether it be to evaluate the volunteers that come in, to analyze the imaging, to analyze the blood that comes in, to recruit, to perform outreach, to educate, to analyze data, and to, at the very end, collect and analyze any brain donations that can be the end of participation for a participant. At the heart of the center is a longitudinal study of nearly 500 participants that are seen on a yearly basis, including up until brain donation. And so we are very thankful to our participants, both current and past, who have really been so generous with their time to help us run our center, to advance our knowledge in this field. We've been very successful over the prior decades as an ADRC, and we have made significant advances to the field at large. We were the center that really was a lead in pioneering the clinical staging of Alzheimer's disease. We identified early stages of it, including the preclinical stage that I'll talk about more, as well as the term mild cognitive impairment, which is a precursor to dementia. We developed widely used methods to evaluate and monitor psychiatric symptoms that are often found or even early in the dementia process. And we helped establish the use of MRI and PET scans to diagnose dementia at early stages, and these are now used at the bedside in the clinic. And so I wanted to now turn to specific projects, specific research areas that we have been advancing in very recent years. And first, I want to talk about the concept of subjective cognitive decline. Before I get to that, I wanted to talk about the stages of dementia and specifically what we mean by preclinical and preclinical Alzheimer's disease. So to the right is the stages of dementia. The 7-stage breakdown was actually established at NYU by Dr. Barry Riesberg. And these stages include normal, the precursor to dementia called mild cognitive impairment, at which people are still very independent, but there are measurable cognitive deficits. And then 4 stages of dementia: mild, moderate, severe, and end stage, where that person unfortunately has increasing dependence on others for their daily activities. And so we know that maybe more than 30% of older adults may be at the mild cognitive impairment or dementia stage. And so this is a growing problem that we need better treatments for and better detection strategies for. And so we felt that it was important to identify a stage between normal and mild cognitive impairment that maybe could be intervened upon, and that's called the preclinical stage. So preclinical means that that person has no objective signs of disease. There's no objective impairment. They test normally on tests of cognition, of thinking. But there are some biological signs of disease, and that person is at risk. So this is a transitional stage between normal and that what we call prodromal or pre-dementia stage of MCI, mild cognitive impairment. And preclinical, this concept of preclinical is in other diseases as well across medicine at large. So where does preclinical fit in with all of the theories of Alzheimer's disease in particular? So to the right is a very famous model of what we know about the Alzheimer's process. And it's a series of curves. So at the x-axis, this is the stage, and the y-axis is sort of the severity of the biological or clinical marker or symptom. And so you can see the first curve that rises, even during the normal phase, is the amyloid curve. Amyloid starts to rise, and that's followed by tau, development of tau. Those are the 2 key hallmarks of Alzheimer's. That's followed by brain shrinkage, so changes in brain structure, followed by memory impairment, followed by functional decline. Now, after decades of various trials, in recent years, we have finally have approved two uh, medicines. These are antibodies that reduce amyloid and can modestly impact the rate of decline during the MCI and mild dementia phase. That's highlighted by this light green rectangle. But you can see that by the time we are trying to intervene upon this early stage of Alzheimer's, a lot of these curves are already at their peak or rising up quite exponentially. And so these disease-modifying therapies, DMTs, don't have as much impact as maybe we thought they would. And so that behooves us to look earlier in the preclinical stage. Unfortunately, we don't have any treatments to prevent progression from the preclinical to that next stage of MCI. And we also know that the levels of amyloid and tau don't fully predict the transition from preclinical to MCI. So we really need a better understanding of this stage, both clinically and biologically. So our center has been really focused on this preclinical stage for decades. That's our center's theme. And so Dr. Riesberg, 40 years ago, he promoted the hypothesis that subjective cognitive decline may represent or may associate with preclinical stage of Alzheimer's disease. So what is SCD, subjective cognitive decline? It's a self-perception that your memory or cognition is declining, but you're testing normally, right? So this can occur quite commonly. And now everybody has a bit of memory concern. It's very common these days. What we're talking about is a significant level of concern. And what Dr. Riesberg found back in 2010 using our ADS's data was that indeed people who have SCD, significant amount of it, do have an increased likelihood to progress to that pre-dementia stage of Alzheimer's. At least looking over a 7-year period, that risk was about 6.7% per year. And that was about a 4-fold increased risk compared to people without those subjective concerns. And this is sort of the data that he looked at to the right. So you can see that this is like sort of a survival curve. So as time goes on, you have less and less people who are remaining in the normal stage in SCD, whereas that curve is a little bit shifted to the right. So you have more survival over time in the people without SCD. There have been more studies to maybe make this symptom characterized more specifically because subjective concerns are so common. And so that there are added aspects to this that may have more tie-ins to biological Alzheimer's, really having memory complaints, that these issues are observed over 5 or less years, comparison to peers, worry about the symptom, having a family member also all concerned about you, and the genetic risk factor for Alzheimer's disease, ApoE4, being present. What we've also found is that it could be preclinical to non-Alzheimer's dementias, whether it be vascular dementia, frontotemporal, et cetera. So it's not unique to the Alzheimer's spectrum. And in the last few years, it's been incorporated officially as stage 2, that preclinical stage, in sort of our national research framework. But we wanted to study this concept further in recent years. We first wanted to know, how do people get to this SCD stage? And what we found using our ADRC data is that indeed there are a subset of people who don't have subjective concerns that end up having it. We followed a group of people over about 7 years, and we did find that there were some people who were quite confident with their cognition, who then experienced SCD several years later, and they did indeed have a faster rate of objective cognitive decline. So they were perceiving something that was declining very subtly, although this was happening in the normal range. And there are risk factors to that, being older, and having some baseline relative weaknesses in tests of verbal memory, like remembering a paragraph after several minutes, or tests of attention and working memory, which include digit span. How many numbers can you remember forwards and backwards, right? We then conducted a series of studies to determine what factors in a person's history or about them can shape their symptoms, either the character of the symptoms or the magnitude of their subjective concerns. And what we found was that demographic variables, vascular disease, vascular risk factors, sleep, and mood, like anxiety and depression, can influence the symptomatology and the degree of concern. And not only was memory involved here, but also non-memory forms of cognition, including attention and what we call executive or organization type symptoms. These symptoms had a unique impact on the brain structure of people with SCD, as we found from our MRI data. And we found that several of these symptoms were exerting their effects independently of amyloid and tau in the brain. That's kind of indicating that there are other pathways to risk in this field. And we know that these are independent risk factors for Alzheimer's disease on their own. We've also been studying how do factors like these influence the rate of progression from preclinical SCD to overt mild cognitive impairment and dementia. And again, we found that these same factors can also influence the speed of change, the speed of decline. That included mood, like I said, anxiety, depression, demographics. We found that persons of Hispanic ethnicity were at six-fold increased risk for that progression. Vascular risk factors like high cholesterol, blood pressure, etc., and poor sleep. And again, we found that that change, that progression to pre-dementia, occurred not only with memory, but also with non-memory forms of cognition, including processing speed, executive function, and attention. We also wanted to look at MRI changes in subjective cognitive decline. And what we found was that there are changes on MRIs in SCD in key pathways related to memory, both input pathways, processing pathways, and output pathways of the memory areas. And that's sort of indicated on the picture to the right. And these changes, based on the type of MRI technique we used, could relate to degeneration of the brain cells. It could relate to inflammation there or vascular injury. So clearly, this is a symptom that is not just someone having feelings, but this is really relating to changes in the brain that are subtle until you look more closely. So that being said, I wanted to switch to our next topic, which is looking at maybe another process that could interact with this preclinical risk, and that is COVID. And before getting to that, I wanted to broadly review the infectious theory of Alzheimer's disease that's been gaining a bit more popularity these days. So we know that the amyloid theory, the amyloid targeting treatments have had some, but modest impact on patients. And so this has really led us to question whether this is the only model, or are there alternative explanations or complementary pathways that might be involved. And so a newer idea has suggested that infections from viruses or bacteria may play a role, and by either harming brain cells directly or by triggering chronic inflammation. And in this framework, amyloid and tau may even be protective from infection, but with time they are triggered to accumulate faster because of overwhelming infection, and they're accumulating faster than they're clear, and that leads to a vicious cycle of further inflammation and brain cell loss, otherwise known as neurodegeneration. And the sources of this that have been looked at are herpes virus, cytomegalovirus or CMV, HHV6. These are very common viruses that are there and at a very high prevalence in the population, but don't normally cause overt significant problems. And so there have been studies, trials even, to look at this theory. But we wanted to look specifically at COVID, because we had the COVID pandemic, and of course many people were concerned about cognitive outcomes from COVID. And so we have been fortunate enough to lead a study specifically looking at how COVID affects dementia risk. And I wanted to point out three major findings from this area of work. First, during the active pandemic, we found that in hospitalized COVID patients, their Alzheimer's blood markers, so we can actually measure, indirect measurements of amyloid and tau in the blood. Those Alzheimer's markers were significantly higher in those hospitalized COVID patients than in people without COVID who even had MCI and AD dementia. So in that state, these Alzheimer's blood markers were significantly higher. We also found, sort of subsequent to the acute pandemic period, that people with long COVID, people who have sort of persistent neurological symptoms after COVID infections, can show brain changes on MRI compared to healthy controls without long COVID. And specifically, there was an area of the brain called the choroid plexus that was both enlarged and exhibited lower blood flow in the long COVID patients. And an example of that finding is shown to the right. You can actually see the long COVID patients having this larger choroid plexus in the upper right, whereas it's a lot thinner in the healthy controls to the bottom. And even recovered COVID patients are kind of somewhere in the middle. So the choroid plexus is a very interesting structure. It maintains the barrier between our spinal fluid and the blood, and it regulates our immune responses. So it sort of makes sense that long COVID is sort of impacting the neuroimmune aspect of the brain, and that might be a risk for Alzheimer's and other dementias. We subsequently found that in the population that had long COVID, that there was an increased incidence of mild cognitive impairment diagnoses, meaning that they were having deficits in cognition that looked an awful lot like Alzheimer's, like mild cognitive impairment. Now, we don't have proof that it truly is, but at least the cognitive pattern was mimicking that. And so I think we're still working on this, and we will have more papers to show you hopefully in the near future. I next wanted to switch to something related to technique development, and that is proteomics. Just to backtrack a little bit, brain tissue has been an invaluable resource in our field. First and foremost, dementia has long been classically diagnosed by autopsy, right? We were often told that, we can't really definitively diagnose Alzheimer's until that person passes away and we look at their brain under the microscope. We now have PET scans that can give us a better direct view of amyloid and tau in the brain, but still autopsy is sort of the gold standard. Brain tissue in this manner is stored in a fixative, and you use molecular stains to reveal the toxic proteins that define the various dementias, including amyloid, plaque, and tau tangles for Alzheimer's disease, and a picture of that is shown to the right. But for a long time, the use of this brain tissue beyond this has been pretty limited, especially in the research space, despite it being a very valuable resource. And so colleagues of ours in the ADRC developed a new method in order to utilize this invaluable fixed brain tissue to study the extensive proteins that are changing in Alzheimer's disease through a method called proteomics. So what does that mean? So at the top is sort of a cartoon of what that process is. Essentially, you take the fixed brain tissue. This is donated by a deceased patient or deceased research participant. And we use a special microscope called an LCM, laser capture microscope, to cut out the individual cells or brain areas that we're interested in. And then we have our colleagues analyze that tissue through a machine called a it's called mass spectroscopy. It's a machine that sort of breaks down the material into all its individual proteins, or called peptides, and then it analyzes the sort of the molecular signatures of all those peptides and identifies them based on standardized libraries. And then we can get a view of all the thousands of proteins that are existent in that brain tissue. And we can compare it to people without Alzheimer's, people who have more rapid Alzheimer's, or people who have different diseases, to compare how the proteins are being differentially changed. And what we found through a series of published work is that there are widespread changes in various pathways related to brain cell connections called synapses, energy metabolism, immune system, that are very known to be relevant to Alzheimer's, and some of them are actually new pathways. And so this has been utilized to show very distinct molecular patterns that could explain differences in symptoms and progression in Alzheimer's disease. And this work has been helping us find new biomarkers and maybe new treatment targets. So one example is a molecule called cicernin-1 that can potentially be used to distinguish Alzheimer's from other tau diseases. An example of kind of the readouts that you get are in the bottom right. So this is analysis of the proteome of a tangle. And you can see all these, a myriad of different biological processes that were determined that were related to tau pathology. And all of these involve different potential treatment targets. I next wanted to switch to our work on vascular disease and dementia. So why talk about blood vessels at all? Well, we know from the epidemiology of dementia that vascular dementia is the second most common cause of dementia, as the pie chart is shown to the right, upper right. And it can also be commonly mixed with Alzheimer's disease. So up to, maybe even a third of dementia is caused, at least in part, by changes in blood vessels, either primarily or concurrently with Alzheimer's. Now, vascular dementia was originally defined as dementia accumulated from individual discrete strokes that are typically from the larger blood vessels in the brain. But as shown in the bottom right, the vessels of the brain are very complex. They include very, very tiny arteries or arterioles or capillaries, as well as veins that dynamically have to respond to the complex metabolic needs of the thinking brain. And we don't really have a very good understanding of how things change in those arteries and veins and how that relates to dementia. So that's something that we wanted to look at as well. And so through a series of studies in our ADRC, we conducted critical work that demonstrated that damage to these tiny brain vessels, arterioles, capillaries, white matter circulation, not just plaques or tangles, can play a central role in cognitive decline, especially in older adults and people with high blood pressure or vascular risk factors. We also showed that such damage produces very distinct clinical and imaging profiles, suggesting that dementia is a combination of brain protein pathology and blood vessel injury. And we've developed techniques to measure such impaired blood flow regulation, vessel wall thickening, and microvascular damage to help in supporting new tests for early stages, as well as potential new treatment targets. So here's an example of one of our papers that was actually looking at veins. We often ignore veins. We always talk about arteries, but veins are important because they are receiving the utilized blood supply and recycling it back again. So this was a study that actually was able to, using imaging techniques, isolate the venous structure deep in the brain that can be a critical determinant of cognitive decline in dementia. And last, I'd like to end with a very hot topic, and that's artificial intelligence. I think we've all been hearing a lot about AI, in the news, and maybe some of it are using it to, make our lives more efficient. But I think there's a tremendous amount of scope to apply AI techniques in dementia, in both diagnosis and even treatment. And so with colleagues of ours in the ADRC, as well as in data science, we have made some very interesting developments in this space. First, we have developed artificial intelligence systems that can analyze large medical data sets, brain scans, electronic health records, and clinical notes to detect and understand dementia earlier and more accurately. And one of our key goals in our papers on this topic is to turn that sort of massive clinical data that I, as a doctor, have to, go through when evaluating a patient into reliable, usable insights that will help clinicians improve diagnosis, predict disease course, and really reduce the manual effort needed to extract key information. And this approach can also allow us to reveal how the AI works. So sometimes the AI gives you the answer, and you don't really care how it got to the answer, you just want the answer. But sometimes we want to be able to, what we call, open the hood and look underneath and ask, okay, how did this algorithm predict that this person might have dementia in the future or might be at increased risk? Where is it looking on their MRIs? What clinical features, what medical conditions, what medications that we never really thought about are adding to that AI algorithm's prediction, so that we can improve not only our predictive algorithms, but this may be a way to develop new therapies and just understand the disease better. And so to the right is sort of a typical pipeline of what one of our AI pathways looks like. This is actually a very recently published study that was trying to detect neurological disease at large, not just dementia, from just CAT scans. And the idea was, CAT scans are really common. People go to the emergency room and get CAT scans of their head all the time for various things. And that is a data set that's there that could really be useful to detect things that maybe we're not necessarily looking for, to help patients proactively. And so this is just a description of the processing pathway of an AI tool. And so I wanted to just summarize the main take-home points of this talk, and this will leave us plenty of room to ask questions and discuss further. So first, we have furthered the idea that subjective cognitive decline may indicate a preclinical stage of Alzheimer's and other disease, defining a symptom whose outcomes are shaped and accelerated by risk factors, many of which are modifiable. Two, we have shown that COVID, both acute, severe, as well as long forms, may be associated with elevated dementia risk. We've developed proteomic technique that has revealed biological pathways that we never necessarily knew about in Alzheimer's disease at an unprecedented level, and that we can be using that to develop new diagnostics and treatment. Revealed using new imaging methods that, beyond amyloid and tau, diseases of small blood vessels can play a critical role in dementia and its risk. And lastly, we have and are applying AI to develop new algorithms to aid doctors in evaluating dementia risk and diagnosis. And so with that, I'll stop here, and I'm happy to take any questions.